When you view cells through a microscope, you will most often see them in a non-reproducing state. The nuclear membrane is clearly visible and inside the nucleus. One or more nucleoli can be readily seen. If proper stains have been used, the chromatin, a mass of long, thin, intertwined chromosomes, will stand out. This non-reproducing stage, in which a cell spends most of its life, is called interphase. About midway through interphase, each chromosome and the DNA it contains replicates. To replicate is to make an exact copy. Chromosome replication cannot be seen directly, but chemical tests show that it occurs. This replication provides a second set of chromosomes. Following chromosome replication, the final events of interphase occur. Then, two identical sets of chromosomes will be equally distributed to daughter nuclei during another part of the cell cycle called mitosis. Mitosis is usually, but not always, accompanied by a division of the rest of the cell to produce two daughter cells. After mitosis and cell division are completed, the new daughter cells enter interphase. Together, interphase and mitosis make up the cell cycle. Act 1. Prophase. The first act among the four stages of mitosis is prophase. Opens with disintegrating nucleoli. The thick strands of chromatin are forming loops and condensing, growing thicker and shorter in the process. Separate chromosomes cannot yet be seen clearly. A little later, the nucleoli have been disappeared completely. Chromosomes become clearly visible, and the nuclear membrane is beginning to break down in animal cells. The two pairs of centrioles begin to separate and migrate toward opposite poles of the cell. Microtubules form between the two pairs of centrioles and become assembled into tiny fibers. These fibers, located between the centrioles, form a football-shaped structure called the spindle. Some fibers are quite short, extending just a short distance from the centrioles. Many of the longer fibers overlap one another at the equator of the spindle. Although most non-animal cells lack centrioles, they do have spindles. Still later in the prophase, the nuclear membrane has completely disappeared. Chromosomes appear even more distinct. The fact that the replication has occurred is noticeable because the chromosomes are double-stranded structures. Each strand is a replica of the other and is called a chromatid. The two chromatids of chromosome, called sister chromatids, are joined at a region of attachment, the centromere. The centromere region of the chromosome serves not only to attach the two chromatids to each other, but also to attach the chromosome to the microtubules of the spindle. Act 2. Metaphase. The short chromosomes become attached to the spindle when the microtubules of the spindle capture the chromosomes by their centromeres. Interaction of the spindle fibers causes the chromosomes to move agitedly as if they were dancing. Eventually, the chromosomes move to the equator of the spindle and become aligned. The dangling chromatids may face in any direction. Once the chromosomes are thus aligned in the spindle's equator, the next act proceeds directly. Act 3. Anaphase. The centromeres split at the beginning of anaphase, the third stage of mitosis. The two sister chromatids of each chromosome begin to separate from one another and move apart. Each one heads towards a pole of the spindle. Although the details are not fully understood, action of microtubules facilitates the movement of chromatids. The microtubules slide past each other, causing the spindle as a whole to become flattened and elongated. The poles move further apart and the sister chromatids are pulled to opposite poles. Each sister chromatid now behaves as a separate chromosome. As anaphase ends, there is one set of single-stranded chromosomes at each end of the cell. Act 4. Telophase. The final stage of mitosis is telophase. Telophase opens as two cells begin to form from one. The plasma membrane of animal cells begins to pinch together at the cell's center as a ring of microfilaments attached to the membrane contracts. Rigid cell walls, however, prevent plant cells from pinching in. In most plants, a cell plate begins to appear, growing outward from the middle of the cell. The cell plate may form from membranes of Golgi bodies or form endoplasmic reticulum. From the cell plate will come the plasma membranes of the two new plant cells. Later, cell walls will form between the membranes. Telophase ends with events in reverse order from those in which mitosis began. The nucleoli reappear and nuclear membrane encloses each set of chromosomes. The nuclear membrane forms from ER, which is endoplasmic reticulum. Meanwhile, chromosomes lose their distinct outlines and once again appear as mass of chromatin. In animal cells, when cell division is completed, the cell membrane pinches together so that the single parent cell is separated into two daughter cells. In plants, the cell plate is completed to form the daughter cells. Thus, mitosis accomplishes the mission. New cells are produced, each of which contains the same set of genetic information as the parent cell.
Cause she hold me down every time I hit her. When I get right, I promise that we gon' live it up. She made me beg for it till she give it up. And I say the same thing every single time. I say you the best, you the best, you the best, you the best, you best I ever had, best I ever had, best I ever had, best I ever had.